Hello, everybody. This is Rafi, and I'm here with my good friend Gabor Erdosi, and he's back on the podcast. Hi, Gabor. Welcome back.
Hi, thanks for inviting me back again.
My pleasure. We've got lots to talk about. I've been following you, of course, on Twitter, like I always do, and there's a lot of new science related to the effect of the vaccines as well as natural COVID infections that I wanted to talk to you about, and you can expand about what you've been saying on Twitter, which has I think ruffled a lot of feathers, like always. But to set the discussion, I thought just before we're recording you were talking about how you go about interpreting research. Maybe you could say that generally, but even more specifically to COVID and vaccine related research. And I thought it was very interesting to set the stage for the research we're going to cover today. So I'll just let you take it away and explain to people how you research and how you interpret the research that you read.
Yeah, I think it depends on time, and unfortunately, I've had a bit more time available than I originally planned for the last 20 months or so. Some of my projects were completely screwed up by the pandemics or the management or whatever. So I had more time available than planned, and eventually I started reading about stuff which seemed timely back in starting in I think in March 2020, and yeah, I'm not a virologist or not even formally a immunologist by training, but what I always do is when I'm have interest in a new area, I start. I do a few PubMed or Google Scholar or whatever searches and try to identify the few reviews which seem to be the most relevant and the highly cited and by scientists I usually recognize from other areas connected to nutrition, metabolism, and immunometabolism. So I had interest in these areas well before. So I was relatively well versed in immunometabolism, and my research on the connection between metabolic diseases and food drove me to better trying to better understand the for example mucosal immunity in the gut, and mucosal immunity is fairly similar in the respiratory system. And also I was reading about the gut lung connection, how antigens are sent around and immune cells are being sent around and this stuff. So it was not far away, and of course I have some basic education, university education in immunology and microbiology and these kind of things. But I think it always starts with reading the basic reviews first, and then there are some areas which eventually pop up, and then you start the deep dive into for example viral transmission or respiratory viral infections and these kind of things. Or for example, how should masks connect into this and these kind of things. And then as always, I start reading more basic research, mechanistic studies, what happens when. And then there are animal models. It's important to understand the differences between the animal models and the humans. And yeah, I think in 20 months you can easily read, I don't know, something like 1,000 studies. So it's a lot of time, and after a while it just becomes more and more complex. So it gets more and more difficult to find the right connections between all this information. And what I can also say about these 20 months is that I've never seen, of course, I think there is always bad science. So if you know how research works, I mean formal academic research works, researchers are under pressure to publish, they spend a lot of their time trying to find the right funding, writing applications and so on and so forth. And then they have to publish, they play some of these games and so on and so forth. So after a while, when you read studies over the years by the thousands, you have an impression on which research groups are almost always fully reliable, and what kind of research you can rely on, and these kind of things. So you can learn to distinguish bad research from good research, to simplify it a little bit. And but the amount of bad research done in connection with the pandemics and COVID and SARS-2 is just astonishing. So I think I've never read so many abstracts and then just throwing away already by reading the abstract that it's a lot of junk science and a lot of distortion as well. And also the interpretations in many studies, even in very high profile, very highly respected also by me, researchers, top tier immunologists, for example, started implementing or including this kind of narratives in the studies, especially in the abstract and the discussion field. So you have to be very careful and pay close attention to the result, to the actual results, and try to strip out all these unnecessary narratives from the discussion parts, because I mean sometimes the interpretation is completely 180 degrees backwards. So, for example, you can often find studies that boosting vaccination on top of natural immunity broadens the immune response and these kind of things. And what you actually see in the data, that this broadening had already happened prior to the boosting, and what the actual boosting does is just quantitatively increasing the response. So there's absolutely no benefit qualitatively, but only just boosting sky high systemic antibodies, which makes zero sense to me. That's not how a recall immune response works. So just a couple of weeks ago, or within the last couple of weeks, I've seen at least 2 or 3 very high profile journal publications peddling exactly this type of we could say misinformation, but if you carefully read the paper, you see what happened, what actually they measured, and what's the straightforward conclusion to anybody who doesn't want to follow a certain narrative. That's difficult.
Right, and a lot of the times the most interesting stuff will be buried in an appendix somewhere, which will have discussion.
Just one example, I saw a case report. Now I have shared it several times on Twitter, for example, a case report about on a breakthrough infection in a healthcare worker in the UK. I think I don't remember the place, and so it was a healthcare worker, quite an average person, I think around 40 years old or something like that. So not an elderly person with comorbidities, and a very high viral load breakthrough infection following full double vaccination. Now this full vaccination is quickly moving to 3 jabs, and then I think eventually next spring you will see the 4th accepted only as full vaccination. Okay, that's a different story. But what happened was that they reported like 1 or 2 sentences that during this high viral load breakthrough infection, actually the antibodies against the nucleocapsid which is a protein within the membrane body, kind of holding together or fixing the nucleic acids, the RNA of the virus. The antibodies didn't really budge. So what happened there? You had to actually dig up the supplemental figures, and there was a very slow increase in N-targeted antibodies, but even 2 weeks into the high viral load infection, this was way below the limit, the threshold where you say that you developed a sufficient antibody response. So then you start thinking that, oh, this is something like a original antigenic sin, when you had a double vaccination, and there is a new antigen introduced together with the old one. The immune response only focuses on the old one, so your immune system kind of doesn't see all the new antigens introduced. Initially, I don't know, a year ago or even half a year ago, I was hoping that a breakthrough infection will extend the qualitatively the immune response. So those who hadn't been infected prior to vaccination at least can benefit some of the infection. Benefit without the obvious drawbacks of severe disease. But it doesn't seem like the case here. And now more and more data seems to confirm it. I heard it from actual doctors measuring antibodies in their patients and not seeing N-targeted antibodies in their patients following breakthrough infection. This is the same on a country level in the UK. They report S antibodies, seroprevalence, and N antibody, and S continues to increase together with vaccination and infection of naive people, but the N antibodies are almost completely flat. So even if you have, these are only driven by new naive infections, the very slow increase. And you have more and more a quickly increasing number of breakthrough infections without a response to other antigens in the virus. And I think that's not really good. And the problem with this is that if the virus manages to evolve in a way that mostly counteracts the spike, the old spike vaccines, so it develops new spike mutations and evasion of these anti-spike immune responses occurs to a great extent, then protection from the old vaccines will be really poor. And what you can do, what they are doing now is add more old vaccines, which is like if you have become obese from highly processed industrial packaged foods, adding more industrial packaged foods into your diet is not the right solution, obviously. So if you have a problem with a single antigen vaccine, then adding more and quantitatively increasing your antibodies, which do not protect you as well anymore, then it's just a stupid idea. I understand that most countries, at least in Western countries and more affluent countries, ordered 3-4 times the amount of vaccines what they need for their population. So like 3 times the number of the population. It's the same in my country, and I know in many Western European or in the US, this is very typical. So what can they do with all these vaccines? They want to use it, so they prescribe a third injection, and then eventually they will subscribe a fourth injection. But the more frequently you administer these antigens, the less time you leave for maturation of immune responses. And I think this is already a very basic problem with the original dosing regimen of the vaccine.
Right, and we'll get to that definitely. So I just wanted to mention a very sneaky thing that's happening, because you were mentioning that the vaccine is not producing a lot of antibodies to N antibodies, which is targeted to the nucleocapsid.
The vaccine produces absolutely zero because it doesn't contain the antigen. So
right.
The problem is the breakthrough infection with the actual virus. That is not producing antibodies against other antigens. That's the problem.
Right. So the vaccine seems to be impairing immunity to the response to the natural infection that one might encounter. There's data suggestive that this might be impaired when you look at the N antibody levels. You would expect them to be higher, but they're not.
Yeah, I think we view it as a impairment, but evolutionarily, this is a proper reaction by the body. So if you imagine that if you are infected with a pathogen which causes systemic antigen dissemination, that's what occurs in vaccination, because it's introduced into your muscle, so it's introduced behind your primary barriers. You have this mucosal barrier and you have your skin. These are the major barriers between the environment and your body, and you straight introduce the antigens behind the barriers, and that's a systemic dissemination of antigens. So it's like a kind of a severe disease.
Right.
And when you have such a dissemination of antigens, and 3 weeks later you experience the same, that must be the same pathogen. So there is no huge evolution taking place on a population level within a population. So you are being reinfected with the same one. So boosting your immune response, with the same antibodies works 99% of the time. So this is just the right response to a evolutionarily proper stimulus. But the problem is that this stimulus is not proper. Because normally, if you mount a successful immune response and you tackle a viral infection, for example, you are never reinfected in 3 weeks. That never happens. I cannot tell you, unless you are, of course, severely, profoundly immunocompromised or something like that. If you successfully tackle a infection, it lasts for months at least, or typically a year or more, at least a season. If these are seasonal respiratory viruses, it lasts a season. So until the next season, which is more or less a year, there is no expectations to see this antigen in high numbers within your body and not at your mucosal surfaces. So I think that's the basic science failure right there. These antigens should be introduced at mucosal surfaces first of all, and if it's injected behind the mucosal surface, there should be I think that the dose should be limited, and also the time between the doses should be greatly extended, because that's kind of mimicking the evolutionarily appropriate immune challenge, so that you have a lower dose priming injection, and then you have a kind of a boosting higher dose injection several months later. I think ideally at least 5 months later, but it could be 1 year, because this is what was already shown, that the natural maturation of immune responses goes on even 12 months later, 12 months post-infection. So your body is still trying to find the best antibody secreting B cells and getting rid of all the others. so that you can mount the broadest possible immune response, kind of preparing for the evolution, what the virus can achieve within 1 year. So in 1 year you will have these variants eventually, and your immune system is perfectly prepared to fight those off with these. It's called affinity maturation of B cells. So it goes on for a year at least, and we should wait until the body refines responses, and then you are not susceptible to any variant. And in this case, I think it wouldn't be a problem that the vaccine only contains a single antigen, because the spike itself is a long enough sequence, is a big enough protein to contain a lot of conserved sites beyond those that can quickly mutate. So I think a spike-only vaccine could be okay. It's not perfect, but with the proper dosing regimen, it could have been much better than what it is now.
Right. And the sneaky part I wanted to point out is that when you run PCR tests, at least in France, I saw the ones that were run on myself, they will be looking at the S, so S protein, spike protein, and at N, nucleocapsid. So when they want to count the cases and make them explode, that's how they'll run the PCRs. But when they're doing the vaccines, they're more interested in just the spike protein and not very interested in the N antibodies. So it's incoherent for me to have a test which will not overcount, but let's say count the cases fully. Yet when they look at antibody responses on which to say, hey, look at how good our vaccine is, they're not measuring N, or they don't seem to care about N. So there's a inconsistency there between the PCR tests and the antibodies that they're measuring. And as you've been saying, look beyond the blood, look at the mucosal surfaces. So there is also the IgA effect at the mucosal surfaces, which is especially prominent and vigorous in younger people, which may explain why even when encountering the virus, they don't necessarily even develop antibodies to it, because the IgA in their mucosa is going to take care of it, not even necessitating this sort of antibody response. So it's really interesting what you point out about the dosing regimen, and also where the narrowly targeted vaccine is introduced, which is in the muscle, which is behind your barrier defenses of the mucosa, the gut, the blood, etc., etc.
Yeah, I think these are the two main issues with the current vaccines. And the third one, I think, is the poor control over where exactly these lipid nanoparticles go. So when you are injected into your deltoid muscle, and people between the ages of 12 and 112 have been injected with the same dose. It doesn't matter whether you are 40 kilos, 12-year-old lean boy, or if you are a 400 kilo obese lady. So I think this is not really good. As lipid nanoparticles, I think they have a kind of a preference for other lipids. So I expect that if you have higher body fat mass, for example, then the distribution can be vastly different compared to lean and small. So I think that the highest risk groups from vaccination side effects must be, by basic logic or common sense, those who are lower weight and low body fat mass, typically like teenage boys.
Which is where we see most myo and pericarditis.
Yes.
Yeah. Yeah, that's a very good point. And it's interesting because I was reading some of the original studies where they were looking at how they developed the lipid nanoparticle. And a lot of the initial efforts they had, it was not taken up enough. It was sort of, break apart basically. And then they developed this new lipid nanoparticle, which I can't remember the name of, but this is what Moderna, BioNTech, and Pfizer essentially are using, which is a very efficacious uptake. So you get these, like you said, you get this distribution, and you can see the images in the rats of these glowing spots. And there was this worry about, are they going to the testes? Are they going to the ovaries? Where are they going? And they had a lot of guesswork to get the dosing right. This is clearly something that I think was not ready for prime time, because you can see in the papers they're sort of saying, well, it seems okay, but we've got a lot of spots in the liver, but we didn't see a lot of side effects, so we'll just assume that's okay. You know, it was sort of very iffy conclusions there, not very reassuring. And I haven't looked at enough of regulatory documents to compare how they would usually assess these things, but at a first glance, at a naive glance, it didn't seem very reliable, the way they were doing this in rats, essentially, because I don't even know if they've done this in humans. I've never seen, you know, radio labeled distribution of the nanoparticles in humans, so I don't even know how that's acceptable. But okay, let's say that it is. I just wanted to go back to the original antigenic sin, because it's a really interesting concept. So for the people who don't know what that is, it's basically your immune system having memory of the first kind of SARS-CoV-2 in this case that it's seen, and it sort of recognizes it by the antigens you know, that are carried. And then when there's a new, slightly different version of the virus, instead of sort of being, quote, open-minded, right, if we want to personalize the virus, it can't sort of adapt to this new shape that it sees, and it just reproduces the same antibodies to it. And that's why we call it a sin, because it's making a mistake. It's essentially trying to defend itself with the old weapons it used against the first version, and now it's unable to handle the second version properly. So that's a sort of a basic way of explaining it. And there's something that came out just today. I think it's Alex Berenson that found this buried once again in the appendix of some UK Health Security Agency surveillance data, that the recent observations that they made is that the N antibody levels appear to be lower in individuals who acquire infection following 2 doses of the vaccination. And that's what you were talking about, Gabor. So this is something that's known to the UK government, that's known to the researchers. So it's a very vague statement. We don't know how much lower exactly. We're missing data to really confirm this, but it's also supported, at least indirectly, by this high rate of breakthrough infection, which is sort of a newly minted term to say the vaccines don't work, which is just a fancy way of saying, you know, the vaccines don't work as expected in terms of transmission. We know that the viral loads are much higher than we would want in people who are vaccinated. They're equally high, apparently, which is, I would not even have guessed it would have been that bad. I would have expected to be lower, but apparently they're equally as high. So this is worrying, and also, you know, telling us that there's even the potential that the vaccines are leaky enough, which is a term to say that the vaccine is efficacious but not efficacious enough, which means it could drive transmission. And the analogy that's been used by people who have a great Substack called the Boricua Gato is that if you have a sufficiently leaky vaccine, what you can do is you can sort of invert the evolutionary trend, where usually viruses get less, less deadly over time. You mount the sterilizing immunity from natural infection, and you also get a reduction in transmission because your body can handle the viral replication, lower the viral load, and this is where you see things disappear, if not forever, at least seasonally. And that's not what we're seeing. We're seeing, you know, a lot of spread from vaccinated people, unfortunately, as you would see spread in people without, who have not been naturally infected. And this is very dangerous because now you're giving the virus time to replicate, which means time to evolve. And this is a very big problem that we've seen in chickens, actually. And it's called the Marek's disease, where these viruses became so deadly, they were essentially 100% fatal. And they couldn't even spread eventually. The transmission was stopped, not because of an intervention, just because it was so deadly to the chickens. And this is starting to receive some supporting evidence from the fact that the case fatality rate, the CFR, when you adjust for the age, gets to about 3 times as worse than it was before, around 1%, 1.1% maybe. And before it was, 90% lower. So despite the fact that in certain age groups, boosting the antibody levels will temporarily decrease hospitalizations and death. That is true. We can say that about the vaccines. Nevertheless, you can see a higher case fatality rate, which is sort of seems contradictory, but it's borne out in the stats. And this is really worrying because now we're getting closer to the winter season. And now we're going to see, the mistakes that we've been doing in the summer, but have been covered up by the fact that this is a seasonal virus, and it's not the worst period, at least in our hemisphere, to see that happen. So this is worrying, and of course we're limited by the quality of the data we have. Like we were saying before, the data quality is really poor, but it's best in the UK and Israel. It's pretty bad in the US, probably purposely.
We see
hidden.
some manipulation already. For example, in Israel, if you have internal sources, friends in Israel, then there was a report I saw that challenged government claims by using airport PCR data, because everybody has go through PCR tests at Israeli airports, and that was freely available data. So they just went ahead and analyzed that data and concluded that something was not right, not really aligned with official data. And soon after, this open database was removed, or deeply twisted or changed or whatever. I don't remember the details, but it was shocking to see how quickly within days they reacted to this challenge. And for example, locally here, I'm based in Hungary, we don't see any reports, nothing. So you have no data. You have these cases, but the testing is low, and you have some deaths with COVID and the vaccinated, and you see nothing. And then at the same time, you have these government TV advertisements, this propaganda that only 1% of vaccinated become sick with or ill with COVID. And this has been going on for 3 months. I mean, the situation must be much more dynamic than that. So if it was 1% in July or August, it's definitely not 1% in August in October. Mm-hmm. So it cannot be 1% all the time, because that's just. And they refer to some studies, but nobody has seen any of studies coming out from this country or this government. So it's it's the scary propaganda. It's politically kind of understandable, because most governments put all their eggs in one basket. That's the vaccines. So vaccines are everything, and we won't need any more lockdowns or whatever if the vaccines work. They even say it in the propaganda that if vaccines work, the country works. So that's how it goes, for example, locally. And I understand, but I think eventually people will realize that the vaccines are not miraculous, and especially not 6-8 months down the road. And adding more vaccines will not solve, more of the same will not solve this problem for the whole season ahead of us. So I'm a little bit afraid that we'll be back to more lockdowns or whatever limitations they come up with. This has never been based on science, just a gradual introduction of some measures and then gradual removal of the measures with the spring. so right. That's it. It's and then they talk about controlling the spread. They do basically nothing, just screw around with these stupid measures. So okay, but I think we have to be careful and more critical of all this information popping up, because there are some hypotheses which are still just hypotheses, and there is and there had been a debate about these prior to the pandemic, and there is still debate about some of these concepts. For example, what to expect from viral evolution, or from also about the basic concept that these viruses become less and less virulent with time. It's also plausible, if you think about it, that the host populations become less and less susceptible with time. So it's an interaction, and it's very difficult to discern what comes from lower virulence and what comes from higher resistance within the population. And I don't think that this debate is over at all. So what we see is a sharp decrease in the susceptible population, and then eventually fatality rates will decrease. I mean, population level, not really the identified infection fatality or case fatality rates, but the overall problem should decrease with time and with the drop in susceptibles. And also what room the virus has on its spike or what level it has to jump to mostly evade vaccine-induced immunity. That's still heavily debated. We are not even sure that this is possible for the virus to do. So we may have a good enough vaccine for preventing deaths and severe disease for longer term. I think it's still possible, but it's really difficult to see clearly because there is no clear data, or we have been following this with a proximity for the very first time. What the proximity we have now, all these genetic sequencing of variants and these kind of things. These variants have always been around of respiratory viruses. So we have basically no clue. I'm sure you remember the 2003 or 4 study in, I think it was a British Columbia care home, where there was a OC43 outbreak, and the only reason they sequenced and followed with PCR was that it was just in the middle or right after the original SARS outbreak, the SARS issues in 2002 and 3. So there was a SARS-like disease in this care home. So a medical group with researchers visited the care home, and they did all the PCRs there, and then they discovered that it was a good old cold coronavirus, the so-called OC43, and the case fatality rate and cases were counted properly, like people with symptomatic disease and the confirmation with PCR. The case fatality rate was something like 8.5%.
Which is massive
among the elderly.
Yeah.
So it's not very much. It's not very much lower than what we saw in care homes during this pandemic. It's I think that was 10-14% or something like that with this pandemic, and that was 8.5. So it's not dramatically different, and that was probably a variant of OC43, a good old human cold coronavirus, and these have been around forever.
Yeah, and this is a good point, because you mentioned the age group, and if people imagine it's kind of like, you can liken it to a risk tree, where the risk is close to zero, very, very low, and as soon as you get the very older people above 70, 80, you see it fan out massively, and the risk just increases dramatically, which 8.5 is pretty scary. It's nearly a 1 out of 10 chance of dying if you develop symptoms. So that's also, like you were saying, similar to what we see with the coronavirus of nowadays, where the risk is really lopsided to the older people. But it's hard for people to wrap their heads around these massive risk differences, and when you have to stratify by age. And since we're talking about the CFR and those death statistics, I think it's worth mentioning again, because this is, like, of course, never discussed in the mainstream media, or even alternative media, that the supplementary data from both the Pfizer and Moderna trials in the 6 months follow-up, they showed no decrease in all-cause mortality. 31 deaths in the vaxxed, 30 deaths in the placebo. And these, of course, they didn't select the particularly old people who are most likely to benefit from it, because this is the market that they were trying to attain in their studies. So we're basing a lot, like you said, we have poor quality data. A lot of this is being, back engineered from epidemiological data that's manipulated or not very prevalent. So the best data we have actually does come from the drug companies, even though they've done shenanigans like stop the control group. So now we don't even have a proper control group because they gave the vaccine to those people as well, which is
study population was
tragedy.
also cherry-picked, so don't
Yeah.
worry about that. It was not a fully representative sample
Yeah.
of
Yeah, definitely. And we also mentioned before the lack of data, like you said, in your country. And even in Canada, where there's better surveillance, which is not great, better surveillance, even when they do collect the data, it's really twisted. And there's a very good example, once again, on the Borica Gatto Substack, where he goes over it. And basically, they've presented the cases of myo and pericarditis, which is inflammation of the, you can imagine this as sort of net around the heart or the actual heart muscle, where you get inflammation. It can give you scarring. It will, it'll decrease your cardiac function in many cases, even in younger people. And if you look at the males who've had 2 doses of the vaccine between ages 18 and 24, they say there's about 103.9 events, so myopericarditis events per million doses. But that's a weighted average. In fact, if they were being honest, they would present that as a sum of dose 1 and dose 2. And when you go through those stats, you see that it's actually 215.5 events per million doses. So to put that into context, because that can seem relatively low, right, in absolute numbers. But if you compare that, so this is, from December 2020 to October 2021, so that's 10 months. If we compare that to the MMR vaccine, measles, mumps, and rubella, over 10 years, there's 168 serious adverse reports. So that's 17 per year. So that's 253 times higher with the current COVID vaccines, if you normalize that to the US population. So the level of serious adverse events, not just I have a headache, but serious adverse events, it's ridiculously higher. It's about 54 times higher. And the normal vaccines aren't even on the market around 1 per million of these serious adverse events. And we're dealing here with like 54 per million adverse events. I mean, this is ludicrous. The number of serious side effects from these vaccines compared to all other vaccines combined, it just outstrips it in a crazy way. And I think we've lost sight of proportion, because we keep hearing relative risk figures or of vaccine efficacy, reductions. That's what they presented in the studies, this 95% magical supposed, efficacy. But if you look at the absolute risks, or the absolute benefit that you get in absolute numbers, it's totally different. You see benefits that range from 1%, 3%, 8%, depending on the age group. The best you get is in the 80-year-olds, where you do get a 24% reduction in deaths, which is a substantial benefit. But in everyone that's pretty younger, it's in the single digits, and sometimes even negative, like -1 in some of the younger age groups. So if this were being presented to people in absolute risk terms and not relative risk terms, I think it would never be accepted. And it's the same numbers. It's just a way of seeing them for what they are. And we know this from statins, how they've manipulated this. So this is an old industry trick that they're using. And it's very sad, because we have so many vaccines that could be given to the right people, and that are being given to the wrong people, putting the people who could benefit from it at greater risk, since we're, like we said, there's this risk that they are increasing transmission, or at least not effectively stemming it. And I think this is a tragedy, because I'm worried that for the future, this will put the mRNA technology at risk, actually. I think there could be a massive backlash. You know how it happens when the research has a bad rap and the emergence of the technology is slowed down essentially for the proper application. And I think this is a real risk that we encounter. And the trust for in vaccines more generally has certainly decreased. I think and that's another tragedy that we're going to have to deal with. So it's a pity, because the mRNA technology was meant for cancer initially. That's how it was originally developed. Actually, they saw that they couldn't make it work for cancer. Now they've transitioned it to the immune system. But as you were saying with your example of dosing and the schedule, they clearly don't know how to use it properly yet. And there's a big risk in backlash from that.
Yeah, I think it's okay to use relative risk for vaccine trials for a certain period at least, because I mean you need to make sure that many people in the study population has a chance to actually encounter the virus, and these relative risk, the absolute risk should add up. If a vaccine really works and there is a high efficacy, the small absolute risk mainly comes from not meeting the virus. So that's the problem of the studies. But eventually, if it runs for example 1.5 years and includes 2 seasons, then during those 2 seasons, most of the participants should have a go, or the virus should have a go at the population, and then the absolute risk reduction increases greatly. But for the long run, and when you already have a huge proportion of the population infected, or just having an encounter with the virus, then these relative efficacies start making no sense at all. Then we should focus on the absolute risk reduction. When plenty of people met the virus, and really the vaccine protected some or a big chunk, hopefully, of them, then let's talk about absolute risk reduction. I agree. It made sense last year, especially because they started the trials in phase 3 in July or something like that, when spread was low, and you have to wait enough time. I actually I think we should have waited 1 season and then see how the vaccines work. So I think it would have been much better to postpone any kind of approval until the spring or so, at least in the northern hemisphere, and then experience what we are experiencing now, that the vaccine efficacy wanes, and the breadth of immune responses are not there. So you can expect frequent reinfections with these variants, even if mild disease with mild disease outcomes. But for example, it could have made a big difference in implementing these vaccine passports, because then you see it makes no sense if the vaccinated spread the virus with almost the same likelihood than unvaccinated, then these passports make no sense.
Yeah, they don't make sense scientifically, and honestly, even if we had a perfect vaccine, I think they would be immoral to discriminate people based on the vaccination status. But like you said, even the scientific reasons aren't there. And let's not forget that those trials, the numbers we're getting are only counting people who have gotten 2 doses. So they don't count the first 14 days. They ignore the first 14 days after your first dose, which is where we see some immune suppression and where we see an increase in susceptibility, not only to COVID, but to any disease, essentially,
Yeah,
because it's having a systemic effect on the immune system.
I'm not saying that the period following vaccination should be counted as fully vaccinated. I'm saying that this period should be monitored separately, and not definitely not attached to the unvaccinated group, because those who receive the vaccine dose are not unvaccinated, even if the vaccine doesn't work yet, and they are vaccinated and without protection. But if you attach them to the unvaccinated group, you miss any opportunity to see if the vaccines may have negative efficacy during that limited time period. And that's actually the few studies and reports suggest who decided to report on that 14 days following a injection completely separately. So there is a 0 to 14 days group, and you see that that's very often negative. In one study from Denmark, and in a in the healthcare worker group, it was more than 100% negative.
Wow. Yeah,
That's not small difference.
no, not at all. So if you could wave a magic wand and change what we'd be looking at, um, what would you be looking at in the blood, first of all? And would there be something we could be looking at in the mucosa to better assess a person's immune response, either to natural infection or to the vaccine?
I think first of all it should be clear, not by now, but it should be clear from decades of immunology research prior to the pandemics, that those who were infected are immune. Infected and survived without severe symptoms, maybe, because if somebody barely survived, they may be really at risk for another severe disease. But those who have survived without severe disease, those are immune, and those are immune for years. That's what basic science research suggests. So questioning this within 1 year was completely nonsensical. So providing the basis of this questioning from peripheral blood tests of circulating antibodies is just another scientific fallacy. It's not how respiratory infections stimulate lasting protection.
It's IgG and IgM in the blood that we're measuring, right?
Yeah, mainly IgG for longer run. And I think this is not how respiratory immunity works. So it's a basic science fallacy, and I think this can only be fed to people who are clueless. Otherwise, those who studied even just basic university level immunology in their life, it's impossible to believe that it's the main determinant of longer term immunity is the amount of circulating antibodies. Why do you need circulating antibodies if you didn't need them for the first time? If you had the mild disease, you developed antibodies, but they took a little role in the actual tackling of the infection. So you didn't need a lot or almost any of these for the first time. Why would you need them constantly in high amounts for the second time? It's I think it's not even logical. It lacks basic common sense.
Yeah, and what? So talk to us about the memory B cells, because that's something that I've looked into, but it's not entirely clear to me what role they could have in terms of Because like you said, they take some time. They take around a year to keep sort of expressing different antibodies and figure out which ones are going to be more useful, especially so that they can counter variants of viruses, which is what viruses do. They evolve quickly. So can we measure it in the blood? Do we have to measure it in other tissues? Is this something that's only available in clinical research settings, or is this something that could be done at scale already?
I think it's only available in research settings, and I think in higher numbers you can only find these in lymphoid structures, so in immune structures like lymph nodes or the spleen or even the bone marrow. We have some limited data, especially limited from the bone marrow, but the little data we have, obviously it's not an easy measurement to take bone marrow samples. That shows persistence of memory cells. So nothing to fear of. But there's a little bit easier sampling site, and that is the mucosal immune structures, where it's much easier compared to bone marrow. At least it's not
Right.
easier compared to a simple blood draw, but compared to bone marrow sampling, it's much easier. And then we have a little bit more data on that. And both memory B and memory T cells persist in the mucosa-associated lymphoid structures. And I think that's really the critical part. And we know from previous research that, for example, memory T cells, tissue-resident memory T cells, which stay around in the first infected tissue, requires antigen persistence or frequent exposure. So if you had a single infection, these won't stay around very long. But if you have a kind of a reinfection, you may not even realize that you were infected, because you can remain completely symptomless. Then it's a stimulus for the same memory cells to stay around, because the pathogen is also staying around. So that's how it works, and it makes a lot of evolutionary sense that you develop long-term memory against pathogens which not just appear once in your lifetime, but they are around every year or every other year. And they come in waves, so-called waves. We have these recognition of all these things which have been always around, that we have a wave of infections. Of course we have, because then there is a different interaction between the host and the virus. So the virus can also adapt, evolve, and the host immune system changes with seasons, with vitamin D, with different environmental factors. So the interaction changes, and we see these waves. We have always seen these waves. At least if you ask an old GP about seeing the waves of patients coming in and complaining about respiratory symptoms, there has always been a wave of infections. That's
Yeah,
pretty normal.
it's pretty normal. And so we've mentioned memory B cells now, and their role in providing us with, it's essentially the wisdom cells. If we wanted to, stereotype them, they give us this wisdom, but we have to give them time to figure it out. And there's also a degranulation response that appears potentially to be weakened by the vaccine, or potentially interfere with the degranulation response that you get after natural infection. And degranulation response, for those who don't know, is when your immune cells can produce a lot of these inflammatory cytokines that are typical, for example, in an allergic reaction, where you get swelling, redness, those sort of things. So it's maybe counterintuitive to people, but a healthy immune response is actually one where you get symptoms to some degree. And when we try to suppress these symptoms, or when we don't induce them sufficiently well with a vaccine that's not behaving as we would like it to, then we don't respond to the disease in the way that we'd like. So maybe you can provide more detail or resolution on that, Gabor.
Yeah, I think this difference in what you mentioned, the degranulation, is already present in different severity infections. So a recent report found that in severe disease, severe COVID-19, T cells responded mainly to the spike, and they had a kind of a little bit dysregulated response with very high cytokine production. And those with milder forms of the disease responded mainly to the nucleocapsid antigen with higher degranulation, and that was the proper immune response. So actually, in my disease, one of the major antigens is the nucleocapsid, and much of the antibodies, even if these are not neutralizing, a big chunk of the antibodies are produced against the nucleocapsid, and there are also a few other non-spike antigens, like one of the NSPs, it was the 3 or 13, I don't remember, and there are even mistranslated proteins which don't really, which phenomenon doesn't really occur in vaccination. But when you are infected with a lot of individual viruses, some of their RNAs are not properly translated, but the translation starts it's called out of frame translation, because it starts with a different nucleic acid. So the whole chain of amino acids will be totally different, because it doesn't start at the start sign, but and not even 3 down, but somewhere else. So you will have these weird proteins, which are some of these are very highly immunogenic, and were shown to induce very strong immune responses. And you notoriously get these in infections, but you don't get these in vaccinations. So in fact, the breadth of the natural immune response is even greater than just the canonical viral proteins. There are these mistranslated, out of frame translated proteins, which also highly immunogenic. So yeah, it's difficult to explain to people who have no basic immunologic understanding. But when immunologists come out and claim that vaccines are of higher quality, provide higher quality protection than an infection, it's straight impossible. I mean, it's
the height of arrogance, really.
You immediately can know that there is some agenda or some funding or whatever in the background, but this cannot be seriously scientific, because it's total nonsense.
Mm-hmm. Yeah, I mean, if there ever was a complex system, it's probably the immune system. So to
it's a
imagine
big
that,
problem. It's a big problem if it misfires. That's what happens in severe disease, but I think there is a good reason what we see, and then the connection between age and certain comorbidities and severe disease start making sense if you recognize that these diseases, these comorbidities, as well as aging itself, are immune-based diseases.
Mm-hmm.
So if you have the perspective that aging is also called now inflammaging, so it's a gradual deterioration of chronic inflammation with age, and it can be delayed, of course, but also obesity related or diabetes related diseases, cardiovascular diseases and other diseases, but also cancer. These have very strong roots in immunology. These are immune diseases, basically. So that's the connection why the misfiring of immune responses are much more likely in those who are old and in compromised in a compromised immunity state. And also that's the case for diabetic people and those living with advanced cardiovascular disease, for example. These are all immune diseases, mainly diseases of innate immunity. And then if you have no proper innate immune function, that's the very first stage of tackling a viral infection. So if you miss out at the early stages, no wonder that late stages go just dysregulated or crazy.
Yeah, and I think since we're talking about, these immune responses, and people have these very poor this poor status of immunometabolic health more generally, right? We have rates of metabolic syndrome in the US that are about 50% with a very weak definition. Probably if we applied a definition you and I could agree on, we could probably capture 3/4 of the population, conservative estimate. So
it was 88%
right.
in 2019 study.
88%. I mean, it's hard to believe, but those are the figures at best that we know them. And we have a lot of people complaining of long haul COVID, which is, I mean, it's difficult to assess because, again, we have very poor data on it. It's a lot of longitudinal data. It's often retrospective. It's hard to pin down, but we already have a term for these sort of issues when we encounter respiratory viruses, which is post-viral syndrome. And it's a general term because it's hard to be very specific. The symptoms vary wildly because the immune system affects pretty much everything. And they present at different moments of the year because our immune systems vary in their performance at different moments of the year, vitamin D levels and whatnot. So tell me what you think about this long haul COVID, and what you make of it, and is this a specific scourge that comes from SARS-CoV-2, or are we just seeing what we would expect from any respiratory disease, essentially?
I think we can go back to minute one of this discussion that I don't see sufficient data to tell the difference between other diseases and COVID, because I think it's more than post-viral infection syndrome. I think there is a post-severe disease syndrome, what we can talk about. If you are hospitalized and make it to the ICU for whatever reason, there is this sequelae of symptoms which last for months. It's not limited to COVID, and it's not even limited to infections. If you have a severe disease and you have difficulties to come out of this, then many symptoms can last for months. Now, comparing only to severe infections, for example, to severe influenza, what we should talk about, what is specific to COVID. Are the rates higher at all than what we observe in other severe infections, for example, in influenza? And the limited data suggests that it's similar. Perhaps long COVID is somewhat more frequent than long influenza, but the differences are definitely not dramatic. And then this is the quantitative nature of the question. Is it more frequent? To some extent, maybe. It might be. We don't really know, but it seems that it may be somewhat more frequent. The other one is a qualitative question, that do we see, are there substantially different symptoms? And the magnitude of the symptoms are different. And that data are even less reliable in connection with that one. So I wouldn't even speculate. Very limited data and very limited properly done surveys, because it's basically just asking patients and following sequentially, asking them on a regular basis if they have still those symptoms. If you remember, there was a there was a survey among children in the UK, and what they found that some of the symptoms were actually more frequent among the non-infected. So that's very very weird. So we should perhaps start talking about long lockdown or whatever caused those weird symptoms among the children, lasting symptoms among the uninfected children. It's not at all a easy topic, but some people go around and throw around these terms, long COVID. You have to protect the children from long COVID, and the limited data suggests that long lockdown is at least as common as long COVID among children. So what we should protect the children from is really just navigating the unknowns.
Yeah, and in any case, whether it's real or not, in the sense that it's as bad as people think, I think the recommendation is going to remain the same. You have to take care of your immunometabolic health, whether you think it's a special case or not. The clinical recommendation remains the same, and that means not being locked in your house. That means being exposed to the sun to get not only the effects from the vasodilation that you get from being exposed to UV rays, not only the vitamin D enhancing effect, but also exposing yourself to temperature gradients, cold and hot, which we know has a massive protective effect on the immune system. So maybe it's time that you start your cold showers, or that you jump in a sauna or do a workout where your body temperature increases a lot. And of course, we never hear any of those solutions that are free, that are safe, that can be done at virtually any age. That are accessible to so many people. So it's a real pity that we focus on these boogeymen, and we don't actually we're not practical about it. We're not giving people the tools they need to actually make a difference. So yeah, I think we have a pretty similar opinion on that. I don't see a special case for long COVID. The best argument for it would be that the risk gradient is very steep for people with metabolic syndrome. So maybe they do suffer more severe consequences long term in that hand. But does it deserve a special name? Does it deserve that we freak people out? I don't think so.
Even if it does, I think that the difference comes from the differences in the virus itself, because at least a good chunk of these symptoms appear to be autoimmune problems. And that's probably why much more women report long COVID than men, because in general women tend to struggle a lot more with autoimmune diseases than men. So if it's different, then it's a difference in the virus, and that's already a great question. Why this so-called novel virus has so many autoimmune stimulating peptide sequences, because that's not how novel viruses after a zoonosis show up. That these novel viruses should look more similar to the original host than to the human host, especially right upon appearance, and this is not what we see. It was already predicted more than a year ago that autoimmunity will be a really big problem, both following infection but also potentially following vaccination, because the spike itself contains like a dozen or 2 dozens of peptide sequences which can be implicated in stimulating autoimmune problems.
Yeah, I remember you sharing that paper that was showing. I can't remember the name of the sequence. It had something with streptococcus in it, where there was something that created a very strong inflammatory response. It was a particular
That was
pathway.
the superantigenic site
Yes.
in the spike. That's a kind of, if you extend the polybasic, the furin cleavage site with the neighboring amino acids, then it adds up to a sequence which is not only very similar to the staphylococcal enterotoxin B, but conformally it looks even more similar. And it was confirmed that with a SEB, the staphylococcal enterotoxin B
Wow.
antibody can bind and neutralize the virus. So it's also very intriguing that you have a uh, endotoxin or enterotoxin antibody developed against a bacterial superantigen, and it strongly binds the viral spike, and neutralizes it. It's rather strange, I would say.
Yeah, I remember that. It was very, 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 very shocking to me how, first of
Yeah,
all,
but
I didn't.
it's partly independent of the autoimmune problems. There was another paper
Okay.
I shared which shows high um, similarity, high peptide mimicry between viral spike parts and human protein parts. But what was even more intriguing, that the same level of mimicry was found with mouse proteins, which is uh, yeah, we already discussed the
Lab
implications
leak.
of that.
Right. Is this, are you referring to the humanized ACE2 receptor or something else?
I think that peptide mimicry requires engagement with the host for a long period of time, or at least
Mm.
many replication cycles. So eventually the virus evolves into a direction so that it looks more and more similar to host proteins, because that's a huge evolutionary advantage. The host won't recognize as many parts as foreign, so it will mount weaker and weaker immune responses. So I think to explain what was found in the mouse, I think humanized ACE2 is not sufficient.
Mm.
I think at least it's not sufficient to explain the simultaneous similarity with human tissues, peptides, and mouse. You need a kind of a chimera to achieve that. And then I think I was thinking along a experiment which was described by who else than Wuhan researchers, and they talked about experimenting with human lung explants. So when you attach
Right.
small lung tissues to the mouse, and then you infect the mouse, I think that's a likely um, origin for that could explain how you have this same amount of peptide mimicry to both hosts at the same time, because it was exposed to both tissues simultaneously.
Very interesting. Very interesting. And yeah, it's the that's I think also one reason why it's been characterized as a what was it, a vascular disease, which really interacts strongly with the epithelium, because a lot of the clotting issues that we see maybe has to do with the fact that it's been exposed to the lung epithelia and has learned to navigate it pretty successfully. So it's not great news for Fauci and Collins, who have a lot of explaining to do now.
Yeah, but it explains a lot of other things. It explains the pushback by many virologists. I mean, if it becomes commonly widely accepted that this was a gain of the result of gain of function research, then what happens with their funding? Even if you are doing just a little bit similar research, and not gain of function research, which many of those scientists implicated in fighting very heavily against this theory. They are involved in gain of function research with influenza viruses, for example, or similar modifications of different viruses. Your funding will dry up very quickly if the public attitude changes. So yeah, I can understand that, but still other scientists should come out and explain what's happening. Not everybody depends on Fauci's funding.
Yeah, no, that's well said. And as imperfect as the US and Europe is, I much prefer to see gain of function research being done there than in China, because the research is going to happen whether we know about it or not. So I think we might as well try to do it properly rather than leave it in the hands of a regime that has no interest in truth, science, or human welfare. So let's hope the backlash isn't going to slow this down too much, and hopefully going to make us take some precautions this time around. Well, we'll see. But Gabor, thanks for your time. It was always a pleasure. It always is a pleasure to talk to you. And we can do this down the road again when we have more, hopefully some better data that comes out. And everyone, please follow Gabor on Twitter. He's at GERDOSI on Twitter, Gerdosi. He puts out a ton of good content. Hopefully, someone like the BBC or the Hill will eventually hire him someday, because their reporting is really terrible on this stuff. So if anyone of those organizations are listening, email Gabor for his CV. Thanks. We'll hopefully do this again pretty soon, and I'll see you on Twitter.
Yeah, thanks for inviting. Bye bye.
My pleasure. Bye.